mentalspod.com, advancing life science research, connecting people and ideas.
We're in South San Francisco at the headquarters of Fluidigm, where we're joined by CEO Gajus Worthington. Today's show is sponsored by Ingenuity Variant Analysis. Identify causal variants from human sequencing data in just hours. With variant analysis, you can interrogate your variants from multiple biological perspectives, explore different biological hypotheses, and identify the most promising variants for follow-up. Welcome to MentalsPod.
Thank you. It's a pleasure to be here.
So, tell us about the gospel of single-cell genomics.
Sure. So let me start with the basic question, which is why? Why would you care about single-cell genomics? And we used to get this question a lot more than we do now, but let me explain the basic premise. So it turns out that individual cells are different. And you can see that kind of on a macro scale because your eyes look different than your skin, and maybe your lips look different than your cheeks. So you know that there are differences between those categories of cells. But it actually turns out that the differences between cells go much deeper than that. That when you go down into an individual tissue, that cells that might even be neighbors to one another are different. This is a phenomenon that's been observed visually in some diseases like cancer for a long time. And the way that you diagnose cancer typically is you take a slice of tissue and you put it on a microscope slide and you apply some stain and then you look at it and you actually can see, aha, that's a cancer cell because it looks different.
Mm-hmm. Plus we hear about the heterogeneity of cancer.
That's right. That's right. The heterogeneity is you can see with your eye in a microscope. That is actually the tip of the iceberg. Those cells that have developed cancer genetically are profoundly different than the cells that they are right next to that are your normal organ tissue. And the differences go even deeper than that. You have cells that become cancerous that are different from the cells that are malignant, meaning that they can kill the organ, or metastatic, which means that they can separate off and spread the disease to the rest of the body, or even these very rare but very dangerous cells called cancer stem cells. You may have heard some about this. They're cells that are incredibly robust. They can withstand radiation therapy, they can withstand chemotherapy, and they can regenerate the entire disease. So within a single tissue sample, you have all these different cell types that are intermingled in one another, and genetically, they are fundamentally different. So what we realized about 5 or 6 years ago is that we had developed a series of tools that would allow scientists really for the first time to start to assess what these genetic differences were and to see how important they were.
So until then, we were just sort of taking a tumor sample or a biopsy and it was
grinding it up.
Grinding it, mixing it all up.
That's right, literally grinding it up. I mean, mechanically grinding it up and then looking at the averaged signal from all of that. So because you can imagine, you have a tremendous amount of background, which is the normal tissue. Then you have these different cell types, each of which is expressing genes in a different way, each of which has different mutations in their DNA, and all that is jumbled together.
Okay, so it's just using averages then.
That's right.
Yeah,
that's right.
I heard a talk recently by Gary Nolan at Stanford, and he used this metaphor. He says, it's like coming in here and putting all of you in this room into a blender, and then coming up with the average.
Yeah, I mean
what would it mean? Right?
What would it mean? Yeah, so another analogy that I use from time to time is like eye color. Like if you average your eye color and my eye color, what would it be? It's an eye color that actually probably nobody has. You tend to have distinct subtypes of blue eyes or green eyes or brown eyes.
So have we all been kind of missing the specificity that we need?
It turns out, so this is the really incredibly exciting and provocative part of single-cell genomics. And the short answer to your question is yes. So in order to understand the progression of let's use a cancer example. We'll stay with that for a minute. In order to understand how a tumor goes from being dangerous but curable if you cut it out to really dangerous, meaning that cells are now metastatic and can separate off and go into your bloodstream. In order to understand that progression, you have to understand why the individual cells turn on some new genes or develop a mutation or two, or maybe it's a bunch that fundamentally change their behavior. There's no way to tease that out from the averaged material. You have to look at an individual cell level.
Okay, so what you're talking about there is the function a cell plays in a network.
That's right.
Or a pathway.
Or the function that a cell plays in driving the biology even. So I'll give you another example. And these are the examples I'm using are real users today. Like we have There's a guy named Mike Clark at Stanford who was assessing this very problem with breast cancer patients and understanding what makes these cancer stem cells so robust, so dangerous, and what could you potentially do to disrupt the pathways that they turn on that are like shields, like allow them to pump out chemicals or withstand radiation therapy. Another example of real scientific work that also is incredibly exciting is HIV. So it turns out that you have in your immune system is fantastically complicated. There are a dizzying number of different kinds of T cells that are adapted to ward off disease. And it turns out that a tiny subfraction of your T cells, literally hundreds of a percent of an enriched population, can be pivotal in understanding whether or not you're going to respond to a new course of drugs or not, if in fact you had HIV. So the only way to, again, you have an averaging problem. You've got this tiny subpopulation of T cells that maybe on the surface look just like the other cells that are around them, but internally they have other networks and pathways that have been turned on that are the difference between whether or not you will respond to a certain regimen of drug therapy or you don't.
So tell us how your technology is driving the study of single-cell genomics.
Well, think about the problem of single-cell genomics. I mean, first off, a cell is infinitesimally small. I mean, I think there are on the order of a quintillion number of cells in your body. So they're incredibly small. So first off, you have to think about doing biology on a scale that is, between 3 or 4 orders of magnitude smaller than the tiniest microtiter plates that exist today. So how do you grab hold of the cell? Once you've released its genetic contents, how do you manipulate those? How do you amplify them? How do you make sure that you're actually assessing the cell itself and not, the soup that's around it? So it immediately makes you think of microfluidics. So if we take the chemistry and the experimentation and take the dimensions down by 3 orders of magnitude, suddenly you're on the scale of the cell itself. So that was some of the early provocative thinking for us. Before it was clear that there was going to be a bunch of great science. This is maybe 2005, 2006. We started to think about, we're actually in a position where we could enable some folks to do genetic analysis on individual cells because we need so little starting material. And the kind of, I guess I should back up for a second. So what we do fundamentally, technologically, our technology platform allows us to radically miniaturize the experimentation. And so I mean take it from through microliters down to nanoliters or even lower than that. And to take all of the surrounding automation, which is done with robots or tubes or what have you, and miniaturize all of that so that on a single device like this, what I'm holding in my hand, you can do tens of thousands of very sophisticated and complex experiments.
So this is like a lab on a chip.
We've gotten to that point. The lab on a chip for a while was had some negative connotations associated with it because there was a lot of hope for delivery, and it maybe didn't deliver on that. But that is a fact. This chip is the equivalent of an entire room full of equipment, but on a scale that is 1,000 times smaller. And even if you had the room full of equipment to do functionally what this chip does, you don't have enough material in an individual cell to manipulate it with all these complex robotics. So going back to the challenges of single cell, you need to make things very, very small. So you can do the chemistry and biology at the level that light presented. The second thing is you have multiple steps. So you need to grab the cell and separate it from its neighbors. You need to maybe isolate it and look at it and make sure that it's intact. Maybe you want to stain it with something to make sure it's a type that gross type that you're looking for. Then you have to break open the cell membrane, and then you have to capture all the genetic material that's there, every last little bit. And then typically you have to do some kind of amplification to it, whether it's RNA or DNA. Maybe there's some steps in between to reverse transcribe. These are all serial steps that normally in a laboratory you'd have a dedicated piece of equipment typically for each one of those.
PCR machine.
A PCR machine, a centrifuge to bust the cell membrane open, something to do some cleanup work after that, maybe a column, and so you've got this.
You're doing all that on here.
All that on the chip. Yeah, so actually what goes into the chip, to the C1 array, which this device here, you take cells that are in suspension, so they're in a tube. That goes into the chip. We have these isolation chambers where they literally grab hold of the individual cells and capture them. And once they've been captured, we can do some basic biology.
Tell us more about this flagship product, the C1.
Yeah,
what is it? What does it do?
It's very exciting. So the major bottleneck associated with single-cell genomics was the handling. How do you isolate the individual cells? How do you break them open, collect their genetic material, and then prepare that for analysis? That was being done manually, torturous, literally with these things I was mentioning earlier, mouth pipettes, very, very difficult, error-prone. The C1 automates all of that. It makes it incredibly easy. You just put in cells that are in suspension, it isolates the cells, it processes 100 of them in parallel, and exports 100 genetic libraries that are then ready for analysis.
100?
100, yeah. So it's a single pipetting step of cells that go in. We produce 100 libraries for analysis. So it's been really amazing to, frankly, to experience the rise of the C1. Nothing like we've ever done has been this provocative to the field and this rapidly embraced. And I think that is really a strong signal for how important single-cell genomics is and how much latent desire there was to really analyze the genetics at this individual, at the fundamental unit of life.
Yeah, I mean, perhaps intuitively, right? Scientists
absolutely
subconsciously, we need this.
Yeah, they need this, but then they look at the workflow, like what would I have to do to actually engage in it? It was like, oh my God.
Prohibitive.
Totally prohibitive. And so what the C1 really democratizes single-cell genomics. It enables with a benchtop system that's maybe 18 inches wide and a couple feet deep.
And the price?
For you, $150,000. That's the list price for the C1. Yeah.
So does this replace the equipment that you would need in the lab?
No, because you're talking about an entirely new field. People that until we
or,
got,
or do you need like a separate bench set up, and this is our single cell bench?
I think it's more that. So we've developed these tools really with a focus on doing single cell genomics because we believe that that's where a lot of the field is going to go. So maybe in the broader arc of time, as more people get into that field and realize that they need to be doing their sequencing at an individual cell level and their expression profiling at an individual cell level, there maybe will be less interest in doing it with aggregated tissue. So maybe there'll be less activity on some of the more conventional legacy equipment.
Yeah.
As a field kind of moves towards single cell as being more of a mainstream approach.
So do you have all the technology for single cell? I mean, you can do the PCR. You can do. Would you do sequencing?
So we don't have sequencers ourselves, but we've developed the machines to be able to prepare libraries so that they can be sequenced on the installed base of sequencers. And that was a significant challenge. So we, as part of what the C1 does, which is our flagship product for a single cell sample preparation, it captures cells, allows you to treat them, then takes it through this multi-step process to then ultimately amplify enough material off the individual cell that you can sequence it.
What has been the biggest challenge?
The biggest challenge has actually not been technological for us. There's a very natural fit technologically. The biggest challenge was actually in the early days of this field was the skepticism surrounding it, that it was cognitively different.
And what would an example of that be?
Let's see. So that could you really see the differences in individuals? I mean, is your technology really that sensitive?
Okay, so questioning the technology.
And
questioning the purpose?
Not so much so there.
People have been sold on that.
Yeah, I mean, I think that more, the bigger, from our perspective, and this has evolved fairly rapidly, particularly in the last year. There is a lot of openness to the notion of single-cell genomics. I mean, people get it very quickly, and a lot of people were thinking about it way before we came along. Mm-hmm. It's more a question about accessibility, and I think that's been the biggest challenge, just really communicating to the field that you can actually do it. It's available. It works robustly.
And how do you do that? I mean, how do you
Well,
overcome this challenge?
it's a kind of a classic a development of a new field in biology. You need to work with really cutting edge, highly credible initial users who are going to do great science. And then you kind of distill it all down to that. It's really about the science.
Mm-hmm. What's an example of an early user you've had who's done something?
Well, I was talking earlier about the cancer example. So Mike Clark and actually Steve Quake worked with them too at Stanford. Were able to actually show what was different about these cancer stem cells, what pathways they were turning on that released oxygen scavengers, way more oxygen scavengers than normal. And that allowed them to withstand more radiation therapy. So what the observation was that you irradiate a tumor, you kill everything, but then in a few years it grows back. What was happening was that these cancer stem cells were turning on these pathways, making them more resilient to the radiation therapy and then regenerating the tumor. So they were able to show which pathways were turned on that were allowing them to withstand the radiation therapy. They were actually even able to then inhibit those pathways and show that if you did the next step then was that you. So this is potentially very profound. I mean, it opens up the opportunity of a therapeutic that would be maybe administered in combination with radiation therapy. That would sort of,
target the specific.
bring down the shields of the cancer stem cell and then hit it with and then wipe out the entire tumor. So here's an example. Another cancer example is Stephanie Jeffries, who's been doing work with circulating tumor cells, and that holds a tremendous amount of promise for diagnosis and also for assessment of the progression of disease. It's really hard to get a needle biopsy of your pancreas. And so one of the great hopes is that if you could really precisely analyze the genetics of lots of cells that are in the bloodstream, you could find these rare circulating tumor cells at an early stage of the disease, and by analyzing them, you could tell what kind of cancer that you had. You could tell maybe also what specific mutations are present that would tell you whether or not to take this drug or that drug. So it has the potential to be a non-invasive and very powerful means of diagnosing and monitoring the progression of cancer. And so Stephanie Jeffries has done a lot of great work showing that she can in fact isolate these these circulating tumor cells and then analyze the genetics at an individual cell level, and that tells you a lot about the disease.
It seems that there could be a cost-saving aspect here, because you hold that up and say this is like in a lab.
It's true.
So as far as all the equipment and the
there
time
definitely
pipetting,
there definitely
and
is. And there's no question. So we reduce the amount of reagent that you require to do these experiments by a factor of 1,000, and the reagents are typically very expensive. We reduce the amount of equipment that you have to have from a room to a single box that sits on. There's definitely a huge cost savings associated with just using the technology, even for sort of mainstream gene expression or genotyping applications. It definitely has that virtue. And maybe that's our next interview perhaps, there's a whole nother application for our technology that's really in very high throughput production applications where that cost is very, very important. But coming back to single cell, I'd like to give you a couple more examples of the science, because I love this stuff. I really do. So another field that has a tremendous amount of promise is what's called IPS, so induced pluripotent stem cells. You've probably done a lot of work around that. And so the idea is that you could take a sample of my own skin and reprogram those skin cells into potentially anything. You could make neurons, perhaps, and heal nerve damage, whether that be something like Alzheimer's disease that comes with age, or God forbid, if I was in a car accident and had some severed nerves, maybe you could manufacture nerves. Or if as I got older, I developed heart disease, I could make heart cells potentially that could treat heart disease. It has tremendous promise. But what's true, and a guy named Rudy Yanich at the Whitehead Institute just published a beautiful paper showing this, is that when you take these cells and you walk them through the process of differentiation from one type to another, they don't do it the same way. You don't get to march everybody in single file. Some of the cells appear gifted They're able to make the transition from one cell type to another very readily. Other cells...
This is after they're already an induced pluripotent
No, this is
stem
when you
cell,
take
or to make it
to make
an
it.
IPS.
Yeah, to make
Okay.
it. Yeah, to make it, and to make it, to either take
Some
it
are
in-
quick, faster learners than others.
Exactly, and that's why I use the analogy of a classroom. You have a large enough classroom, you got some really gifted students there, and those are kind of like these cells that are able to make the transition from one type to another very readily. Other cells get arrested in their development, and they get stuck, and they're not able to make the transition, and so you have to understand this to make IPS achieve its promise, and the promise is amazing. You have to understand what are the mechanisms by which these gifted cells go from one cell type to another, and similarly, what are the mechanisms by which these cells that get arrested get stuck. And
So you have to look at them separately.
you have to look at them separately. If you jumble them all together, you'll never see the signature of these rarer gifted types, and you'll also really not see the signature of these rarer, really arrested types as well. So we're one of the most fastest moving areas of single cell genomics is in stem cell biology. Understanding how to do IPS, understanding developmental biology. We all come from one cell.
I'm having a hard time understanding why, if you had the technology to do single cell, you wouldn't just convert to that for research.
Well, it's like any new field that goes through the evolution of early adopters, and one of the things that I think the field is very excited about now is the rate of publication. So we have on our platform now we have over 65 publications that are single cell focused, and I think that that's what I said earlier. It's really about the science. So as more and more science is done, we believe that this field will become more and more pervasive. And I think the other thing that's true is that there's still a lot of researchers out there who don't understand or just don't know that it's as accessible as it is, that you could just do it, because as I said earlier, the idea is not a new idea, but the technological challenges associated with doing single cell genomics, I think until very recently, were viewed as so extreme that it probably wasn't going to be accessible. So I think, let me make a kind of a blanket statement here, though, that what we've seen in nearly every tissue type, even muscle tissue, which I would have figured muscle tissue is pretty homogeneous, right? And I got sharply corrected by a guy at Albert Einstein University who said, no, actually you have hypermetabolic muscle cells that are very different from their neighbors. And those hypermetabolic muscle cells, can be linked to your athletic prowess, but perhaps are also linked to the rate at which you age. Because they're hypermetabolic, they're more susceptible to point mutations or structural changes, and those hypermetabolic cells could actually be the source of cancer, for example. So he said it's very important to actually do single cell genomics on muscle tissue. Anyway, every biological system that we've been exposed to, and where scientists have now plumbed the heterogeneity, they've found it, and it's not just interesting and stochastic, it actually drives biology, or raises another set of questions that have to be contended with. So with that as a backdrop, we believe that over a period of years, it'll take years, that single cell genomics will expand to pretty much every area of cell biology and genomics.
It makes absolute sense. I mean, there's this ongoing trend toward, going in closer and closer and more complex, and we hear a lot about, the network pathways.
That's right.
That are important to understand with these complicated diseases.
Yeah.
Even disease classifications, right? We're saying to call all this cancer is just really wrong. There's HER2.
That's right.
Even to call it just breast cancer is
Yeah.
wrong. Susan Desmond-Hellmann was saying, last week, it should be called HER2 cancer or this kind of.
Yeah, absolutely, and you're talking
And you're
about
doing the same thing with sales. You're saying this blanket thing just doesn't really work anymore. We're to the next level. So I'm curious, do you guys have a monopoly on this whole single cell technology thing?
No. I mean,
There's a lot of people doing it.
There was a lot of because it's important enough for people who are doing it manually. There are tools that have been evolved over long periods of time to literally They're called mouth pipettes, and it sounds strange, right? Well, it turns out that you can modulate your breath very precisely, much more than you can modulate, how you're pinching your fingers. So you can use your breath in conjunction with a pipette you can drive the pipette up to the edge of a cell and suck in just a little bit of air, grab hold of the individual cell, pull it out, and then drop it into a little tiny well. And so there have been a number of publications where literally by that manual methodology, scientists have made some great discoveries, but
you have the automated.
Yeah, the field was never going to advance if you didn't solve that problem. It's too difficult. It's too error prone. It takes too much time. So there are definitely folks out there that have done it manually and continue to do it manually. But
so I think you're the next big Illumina then, really. I mean, because your technology is going to enable all this.
What I'm saying, we're the first Fluidigm. Okay. We don't have any aspiration of being, really, frankly, the next anything, but we are absolutely committed to delivering enabling and elegant tools for single-cell genomics. We're in a leadership position right now, for sure. I dare to say, we're the technological leaders. We're the market leaders. We think this field is going to be profoundly important, and it's our mission to provide the best, the most enabling, and the most elegant tools to the scientific community. We think that if you stand way back and ask where can this go, so the kinds of things that you do with tissue right now at a cell biology level, there's a whole myriad of things of culturing and staining and provocation and induction and what have you, and then you take those tissue samples and then you have a whole set of means by which you can analyze them. You can look at protein expression, you can look at gene expression, you can look at genotyping and point mutations, sequencing and DNA methylation. So we think that all of those combinations of workflows are going to be important at a single cell level, and that's what we're committed to enabling.
I totally get that. Now, you were here in the beginning when the company was started. You're a co-founder with Stephen Quake. He had the microfluidics technology from Caltech, his lab. And so, I mean, did you have any idea back then about how single cell science might take off?
It's interesting, and I still have like the original business plan that we wrote, and it was a table not unlike this. We sat down and wrote it out, and I still have the original slides that we developed to start pitching the company. And one of the things that we were very interested in was doing single cell manipulation. We we had demonstrated a chip that could do cell sorting, kind of similar to what at a very small scale what a fax sorter could do. But the thing that we thought would be really cool is if you could sort the cells by surface markers and then do genetic analysis on them individually. It took a long time.
Well, I can see you're really excited about this. And
it's hard not to be.
It is very, very exciting. I mean, every week you probably hear about new things that are being done with it.
It's true. I mean, if you'd asked me a year ago, would we be here now? Honestly, I don't think I would have hoped it, but I don't think I could have imagined that the field would evolve as much as it has in the past year. Where just the other day, I was with one of our customers and one of our users, and they're explaining a twist on single-cell genomics that I hadn't realized before. And so let me explain it. I'm sure you're familiar with systems biology. This notion that's about 10 years old, maybe even more, that if you can apply stimulus to biological systems and then take measurements, you can start to design and map out the genetic pathways and the circuits, if you will, that are inside the cell. Well, what happens is that when you stimulate a system, you have cells that are doing different things. You have cells that are detecting the stimulus and then activating pathways internally, and then signaling other cells, and then they're reacting to that, and then maybe then there's a cascade effect. When you look at the tissue, you're aggregating all those things together. And so you develop a model of the circuit, which is wrong. There's actually several distinct circuits. One pathway is a response in the initial signaling to another cell. The next pathway is another response to that, and maybe a cascading effect. And the only way you can see those is single cell.
Yeah, well, this is the beginning of a series we're going to do on single cell genomics. I really look forward to learning more about it and following you guys. Thank you for taking the time.
My pleasure.
Today's show is sponsored by Ingenuity Variant Analysis. Identify causal variants from human sequencing data in just hours. With variant analysis, you can interrogate your variants from multiple biological perspectives, explore different biological hypotheses, and identify the most promising variants for follow-up. Tell us what's on this chip.
A lot.
I can hardly see anything on there.
Yeah, well, everything is extremely small. But on this chip are roughly 30,000 individual valves. There's a couple thousand pumps, over 100,000 little segments of pipe, and about 20,000 reaction chambers.
So the Bellagio can hire you guys to do a fancy fountain.
So we look for analogies to try to explain how much plumbing is on a single device that's a couple postage stamps inside. So in fact, we dug up some plumbing, like how much plumbing, we asked ourselves how much plumbing is in a hotel room, and then how much plumbing is in a big hotel. And if you added every piece of pipe that is welded or bolted, every valve that is part of fire suppression, the sewage delivering water, even moving gas, natural gas around in every reservoir. So think of everything that could hold liquid in a hotel. You need a hotel that's about 1,000 rooms to have the same amount of plumbing that I'm holding in my hand right here.